Hello and welcome to Nikolai's genetics lessons, and today I have prepared two multiple choice questions for you, and every time I prepare two multiple choice questions that means that questions are easy. So the first question is, which of the following is not a heterogeneous base in RNA? And here's four answers to choose from, and the answer that we are going to choose going to be thymine. Cannot find thymine in RNA because in RNA thymine would be substituted with uracil. So uracil. And second question, DNA synthesis, that is the addition of new nucleotide, is always proceeds. And here is four answers to choose from the promoter, and this is not true because promoter just promote gene translation but do not participate in addition of new nucleotides. In A, the direction, and this is only half true. I will return to this answer. In the 3 to 5 direction, in the 5' to 3' direction. And imagine that here we have a replication bubble and two strands separated. Somewhere here in the middle there was origin of replication, so to this strand of the DNA a new primer would be added, and for example if this primer would have 5' end here and 3' end here, the growth would be continuous from 5' end to 3' end direction, and would be discontinuous in the other direction. It still would be in the same direction from 5' end to 3' end, but we would call such strand, such direction, as lagging strand. So as you see, directionality of the growth would look like in both directions, but actual direction of the growth always would be from 5' end to 3' end. And the same picture we can see on the other strand of the DNA. So if primer would be added in one direction, so here we would have 5' end and 3' end here. So from 5' end to 3' end, it would grow continuously. So this is going to be leading strand, and relative to this origin of replication, the same strand would be lagging strand. So another primer would be added here. It also would have 5' end here and 3' end here, and once again the growth would be to the center, but as you see, we would perceive it as a growth in both directions. Those once again the growth would be only from 5' end to 3' end. So the same strand of the DNA, new strand of the DNA, can be leading strand and lagging strand. Leading strand and lagging strand, just in relation to the origin of replication. Some people who watch pictures in the textbook where only replication fork is shown have wrong impression that one strand is leading, another strand is lagging strand. Actually, every new strand that would be synthesized in the new DNA would be leading and lagging, leading and lagging. And one more comment about promoter, because some people may still think that promoter have to be somewhere at the beginning of the gene, so and new strand of the DNA have to grow from this direction and in the different direction. But actually promoter promotes transcription of the gene, and that means that new RNA strand would be made from the template strand of the DNA. So our question about DNA synthesis, not about RNA synthesis. So that's why we also exclude this answer. And by the way, sometimes promoters can be very, very far from the gene. For example, if this is going to be strand of the DNA, if we would have, say, a gene somewhere here, promoter can be very far away from it, 10 of thousands of nucleotides far from the gene itself. So usually in such cases double-stranded DNA can make a loop, so this promoter would be close to beginning of the transcription. But one more time, promoter helps to promote expression of the gene, and gene expression would be through making of a new RNA. Usually this is messenger RNA, but sometimes this RNA can be structural. Such RNA, for example, we can find in the ribosome. And once again, our question about DNA synthesis, not RNA synthesis. So this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.